Good evening, everyone, and welcome to tonight's Thoracic Oncology Group webinar entitled Beyond the NGS Report, Practical Interpretation for Lung Cancer Care. My name is Ben Kong, and I'm a medical oncologist at Prince of Wales Hospital in Sydney, and it's my great pleasure to chair tonight's proceedings. We have a fantastic lineup of speakers tonight, including three pathologists, Associate Professor Carolyn Cooper, Dr. Jennifer Borofsky, and Professor Stephen Fox, as well as two medical oncologists, Dr. Lavinia Tan and Associate Professor Melinda Itchens. Before we begin tonight, the Thoracic Oncology Group of Australasia would like to acknowledge the traditional custodians of the lands across Australia and pay respect to elders past, present and future. Toga also acknowledges Maori as tangata whenua of Aotearoa, New Zealand, and as treaty partners with the Crown as agreed in Te Tiriti o Waitangi. Tonight's webinar would not be possible without our sponsors, and Toga gratefully acknowledges support from ISI, Merck and J&J for tonight's webinar and their support for the Thoracic Oncology Group and all of our patients. Just to remind you of Toga's mission tonight, our vision is to eliminate thoracic cancers as a source of death, disability and suffering. And through that, our mission overall is to prevent, treat and cure thoracic cancers through research, clinical trials, education and advocacy. And so in the spirit of that mission, we are very pleased to deliver tonight's webinar. For those of you who are not already Toga members, we represent the full range of professional disciplines involved in caring for patients with lung cancer, clinical trial professionals and consumer representatives. And membership to Toga is open to anyone interested in thoracic cancers. So we invite you to join us as we conduct research to ensure patients receive world-class care. Tonight we have a full program which will take you right from the beginning, starting with patients at their diagnosis with an overview of molecular testing. We'll have an overview of how to read a lung cancer molecular report and translate those results into treatment decisions. And finally, outline some principles of molecular testing at progression to assist you with managing your patients. We will have a panel discussion and Q&A at the end of the webinar, and we invite you to enter your questions into the chat on the right, and those will be addressed at the end. To begin today's proceedings, I would like to introduce Professor Stephen Fox, who is the Director of Pathology at Peter MacCallum Cancer Centre, and he's also associated with the Collaborative Centre for Genomic Cancer Medicine at the University of Melbourne. Stephen has research interests in diagnostic and predictive markers of protein and nucleic acid assays, and tonight he is going to give us a presentation entitled Molecular Testing at Diagnosis. Thank you, Stephen.
Well, thanks very much, Ben, and thank you to Toga for the kind invitation to chat to you tonight, and thank you for the audience for pitching up in the evening after a long day. So I'm going to cover what is termed the molecular testing at diagnosis, but what I'm really going to cover is four sort of broad areas at quite a high level. Understanding the fundamentals, reviewing the classification and the clinical implications of findings in molecular testing, talk a little bit about the clinical testing platforms, and probably go into a little bit of the detail there under the hood, so to speak, in the laboratory. And then I'll finish up and try and persuade you that RNA is as important as DNA, and in some circumstances more important in the design and execution of any of your assays that you're going to be using. Just to highlight that there are molecular testing guidelines that have come out from the RCPA, which I do believe you've been highlighted to you, but I'll just underpin that much of what I'm going to talk about is around there, and the entire webinar is probably geared to this sort of level. So what about the cancer genomics fundamentals? I don't want to labour the point too much. You'll know quite a lot about DNA, RNA, and protein, but I just wanted to highlight that in the lab we can analyse all of these analytes, and they have pros and cons depending on what you're trying to find. The other thing I wanted to highlight is that, of course, that most DNA is not actually translated into RNA. Only about 1.5% of it is, and indeed only about 2% of RNA is actually transcribed into protein. And that's because a lot of it is sort of regulatory stuff, particularly sort of microRNAs, long coding RNAs, etc. And so when we're looking at these sorts of things, take that into account. Well, I'm going to start really talking about the types of alterations that you'll be seeing in your reports, and the other speakers down the track will be dealing with these, but I'm going to try and explain them at a fundamental level. So first of all, we have our point mutations, sort of single nucleotide variants, and that's basically you substitute a single codon within the DNA, and I've just given a red to bed in this particular sentence, and this alters an amino acid. Now, it can result in what is known as a synonymous change, that is, normal protein doesn't actually change, or a missense where you actually alter the protein. And the examples in lung cancer are the EGFR L858 and KRAS G12C. And that's where you're changing a single codon, you get a missense new codon, and both of these become activated and thus become oncogenic. So that's an example of a point mutation. If we move into small indels standing for insertions and deletions, these are anything from 1 to 20 base pairs, insertions and or deletions. And the example here is basically you lose the word his there and everything gets shifted, if you like. Now, when it's in frame, it tends to lead to an activating mutations. The examples of the deletions I've given you is exon 19 in EGFR and the insertions in exon 20 in EGFR. Now, the next sort of variant type is structural variant, and this is more of a class, if you like, and it's defined by any rearrangement larger than 50 base pairs, and that's based largely on short-read sequencing, which we started off in the laboratory as just a definition around that. And this is about rearrangements, inversions, translocations. They can be deletions or insertions. You can duplicate segments, and that leads to complex shuffling of the genome. And that can be very small, but 50 base pairs, or be as large as an entire chromosome, affecting one or both. And the example here, I've given you an inversion, where an entire segment has been flipped. And this, in this particular case, has caused a fusion. So that's an example of a structural variant, where you've joined two chromosomal segments to form a chimeric protein. And this sort of, if you like, in this particular instance for ALK and ROS drives growth through kinase. And it's constitutively active as a kinase, and that's why it drives proliferation and cell survival. The next one that's relevant in the lung space is splice site variant, and the example I've given there is a MET exon 14, and basically what you've done is you've lobbed out an entire exon in this particular part of the sentence. So it's skipped over, it's still in frame, but you get a shortened protein. And what happens in exon 14, you lose an element that actually degrades the RNA and then the protein. And of course, if you don't degrade it, you get heightened signaling. And then the last one you'll know about is obviously copy number in amplification. And the examples there are MET and ERB2. And they're basically, you've just got increased numbers of copies in the chromosome. So I also just wanted to highlight the difference between a driver and a passenger mutations. You would have heard about these terms. So a driver mutation confers the selective growth advantage, whereas the passenger accumulates by chance. One promotes tumor development, one doesn't have a selected advantage, but it does lead to other indications that you can use genomically. So you can get a mutational signature out of them and actually understand what's causing the tumor in some circumstances. Very useful in pathology. The other thing is it can become a driver in certain contexts if it affects a particular gene. Whereas the driver mutations are the ones that you all know about and account for 50 to 60% and lung adenocarcinomas as a targetable mutation. And the other thing just to highlight is that these are currently across mutations, and these are not recurrent across patients. Now, those are single mutations, but there's much more interest now in coming into what is known as co-mutations. That's where you look at secondary mutations that are also present in the tumour. So the examples here I've given you are EGFR and TP53, which is associated with the worst progression-free survival on EGFR TKIs. Mostly second generation TKIs, and it's a little bit less clear with third generation. You can also get associations with small cell transformation, so EGFR plus RB1 or P53 or PIK3CA. These are all associated with an increased risk of small cell lineage switch on progression. Another example of a co-mutation is STK11 with KRAS, and this is where you get a reduced immuno-oncology response with chemo, and so the benefit is actually less in patients that harbor this particular mutation with KRAS. There are others like KEAP1, which are also associated with a reduced IO chemo benefit, but they go through a different mechanism. And then lastly, in this particular area, you get MET amplification, where you get, again, acquired resistance and a reduced benefit from your co-mutation in this particular circumstance. And then associated with, slightly similar to the small cell transformation, you get an aggressive biology associated with RB1 and P53 at baseline. This is not an emergence. This is actually a truncal mutation that's formed early on in its development. Now, the other thing to bring in is sort of clonal mutations versus subclonal mutations. So we've discussed very briefly single mutations, co-mutations. An example here is I've got EGFR L858R and P53 as your truncal mutations. But even within this particular tumour, you'll have different clones, so-called subclones, that account for small proportions of these overall tumours. So the clonal mutations are generally present in all tumour cells compared with only a subset of cells. And that can be reflected in some of the reporting. So you get a high variant allele frequency, greater than 40%, in your tumour result, if you're looking at a particular result. And then subclone will be much lower that because only a proportion of the tumour cells will have the clone. Now, if you look at treating this particular patient, you can actually get subclone emergence. So you're killing off with your drug, your truncal mutations, but that's allowing these subclones to then sweep out or grow out, if you like. So examples here are MET amplification, which bypasses EGFR to increase signaling. You can get on-target blocks. So this particular mutation, 797S in the EGFR, actually stops osimertinib binding, and therefore you get resistance. And this particular example is small cell transformation, this particular background. And these subclones often go to different areas within the body as metastases. Now, you've got your variants, you've got your mutations. How do you classify them in the report? And the example I'm going to bring through is the V600E in BRAF. So the first thing to do is to decide whether the variant is actually oncogenic. So we do have a framework for oncogenicity assessment. So you have the oncogenic, likely oncogenic, oncogenic. You're not sure whether it's oncogenic, and then the benign varieties, likely benign and benign. And then having decided whether it's oncogenic or likely oncogenic, you then want to assign a tier of clinical significance. So these are using the ASCO-CAP AMP guidelines. So you have different tiers, 1, 2, 3, 4, which I'll go into a little in a moment. And they're very much around the clinical significance in therapeutics, diagnostics, or prognosis. This is for tier 1 and tier 2. Just to say, which I'll speak about in a moment, there is a new tier within the tier 2 coming, which is level E, which will be coming on in due course. So how do you decide whether a particular oncogenic variant is has the evidence to have clinical significance? Well, you look at a variety of different domains. So you can look at population frequencies, for example. So if it's present in the general population, it's likely to be benign. You can have segregation data. So does the variant correspond with diseases in families? You can come up with algorithms that can predict from the different types of variants whether they're going to change the structure and therefore be pathogenic or oncogenic. Often disease association, does the variant associate with a particular cancer type, for example? You can put it into a model and see if there's a variation in its function. And lastly, of course, there's very important information in the clinical domains. Is it associated with particular types of tumours? Now, having looked at these different types of evidence, you can then assign a point score to the evidence, depending on how strong the evidence for these different domains are. So I've given some point score here of V600E. So I've given the supporting evidence of 1 point for population. For this particular supporting evidence for this Rebel store in silico, there's a point there. It's known as a cancer hotspot in the disease association. Functionally, it's constitutively active, and then you've got 4 points because there's a lot of clinical information. And having done all that, you sum your points, and that gives 12 points. You then look at your table and see whether your score, 12 points, is over the above 6, and 6 and above, particularly likely oncogenic or oncogenic, and this is obviously oncogenic. So you've assigned the oncogenicity to this particular variant. So that's the first part of the puzzle, if you like. And the next thing to do is, of course, decide the evidence strength so that you can then assign a tier to determine its clinical significance. So this is the tiering that I mentioned earlier, the tier 1, tier 2 of the clinical significance. Tier 1 is strong, tier 2 is potential, and here we've got tier 3 unknown, or tier 4 benign, or likely benign. Now, I did mention that there's a new tier coming along, which I'll say something about, but I just wanted to highlight that the same variant is very context dependent. So you can have a KRAS G12C, which is tier 1A, very strong clinical evidence in non-small cell lung cancer, but in pancreatic cancer, the same variant has a much lower tier of evidence. And obviously, this is a movable feast depending on the amount of clinical information or other information that's available. Now, just to go back to the gap that I mentioned, what's been noticed applying this particular classification is that you get some variants that are not strictly tier 2, but you do have functional evidence of significance, if you like, but you don't have the clinical evidence. So you might have the animal models. Similarly, you might have an unknown clinical evidence, but functional evidence. So it sort of stands between tier 2 and tier 3. So they have brought in this level E, which will go somewhere below the level D in the tier 2 area. And the idea is to try and make them available for other clinical trials like basket trials or expansion cohorts. And the idea eventually is that accumulating evidence will upgrade to tier 2. I just wanted to mention that this particular framework is just one of many that you might come across in other particular tests, in other jurisdictions, other types of classification. And I've just sort of listed a little table here, but there is the European one, which is known as ESCAT. And this is what the result. This is a report, which I won't go into because Jen and Caroline are going to show you some reports and take you through those. I did want to mention very briefly some of the clinical testing platforms that are available. So these are very broad groupings and each have obviously principles and different types of alterations that can be detected. So it's very much horses for courses, depending on what you're particularly interested in. Some tests will pick up most of it, but other tests will only pick up small amounts of changes. So for example, NGS panels can be quite comprehensive, but PCR approaches just tend to be known hotspots, SMVs, for examples. The input requirements often are very different, and that can be determined by the types of samples you have. For example, E-buses can be very small and you don't get much DNA, so you might want to choose an assay where the input is very, very low. And then if your turnaround, of course, is very important, you might want to go for a fast turnaround test rather than a longer turnaround time. So they all have advantages and disadvantages. Just going on to not to forget about immunohistochemistry, we do still see great value in biomarkers here. And of course, you will have known about ALK, ROS, and PD-L1. So ALK is an FDA-approved companion diagnostic in some jurisdictions, not in Australia. Quite a good test and has strong correlation with the genomic test. However, ROS1 is the opposite. It's sensitive, but it is not very non-specific. So again, each of these has advantages and disadvantages. We know that PD-L1 is flawed, despite what it says on the label. And then there are others coming down the turnpike, for example, against MET for some investigational therapies and TROP2. And I just wanted to highlight that because the actual quantitation of TROP2 is now going to an AI algorithm based on digital pathology whole slide images. So again, at a high level, we've got the PCR-based methodologies and the FISH. And just to say, for the FISH, you need basically single tests for each different change. But the types of changes require a different type of FISH. So just to be aware that not all FISHes are the same, that there are types of FISH like break apart, fusion, or copy number that give you different readouts. And again, in the PCR-based methodologies, there are differences there that if you're using a particular test, you should be aware of. Again, I'm not going to labor the point. I believe you're going to get the slides. In the interest of time, I will move on a little bit. I did want to mention very high-level target enrichment in NGS workflows, because certain types of tests can do some things and not others, and you should be aware of the difference between amplicon and capture-based testing. So amplicon-based is PCR-based, and these are generally quite good for small panels. And this is the principle. Basically, you have primers against your areas of interest, and you basically get loads of reads based on PCR here. And these are only good for small panels and well-characterized variants, so good for lung, you could argue. They're generally low cost and they're rapid turnaround time, again, good for lung. And they're good for degraded DNA with low input, again, good for lung. But it has disadvantages which you might want to know about. So it has some problems with certain types of nucleotides in the DNA. The allele frequencies are generally unreliable, so don't put too much store in there. And then if you have a primer problem because you've got a SNP over a particular primer, you'll get dropouts, so you won't always get the read. Contrast that with a probe-based, capture-based approach, which can be much more comprehensive and can pick up novel or unexpected targets. It's slightly higher cost and there's a longer turnaround time, but you do get additional information, particularly around signatures and things like TMB and MSI. The other thing it can pick up is copy number, which amplicon cannot, and also some structural variants, which again, amplicon struggles with. And the last thing I just wanted to mention about the power of hybrid capture is just to mention that they can get, if you like, copy number across the entire genome. And that's based on the probes that you've designed actually also bind to all over the genome in quite a uniform manner. So not only can you pick up quite detailed information about your gene of interest, in this in particular instance, EGFR, but also using this off-target reads and then binning these sparse but uniform reads across the genome, you can infer copy number. So this particular example, I've got a gain of EGFR, but you can also detect a loss of, in this particular instance, TP53. So again, you might see these readouts based on copy number to try and give you additional information in comprehensive genomic profiles. And lastly, just to mention that a test is not a test, that there are different types of sequencer platforms, different pipelines, and different reporting systems. And you can have exactly the same DNA going in, and you're using these different platforms and different pipelines, different reporting systems, and you can get a different result out. So just be wary, know your test, and trust your test and your pathology laboratory and get to know them. Lastly, I'm going to persuade you, I hope, that RNA has value when you're thinking about the type of variant you're trying to detect, particularly in lung cancer for fusions. So when you're designing a DNA capture-based panel using DNA as your input, you can detect fusions, but they have to be identified and known. So this particular probe set I've designed against exons and some introns where I know that there's going to be a break apart in there and a fusion to another gene, gene A. And I can detect that using what is known as the tiling across the exons and the introns in this particular circumstance. However, if I've got a break in this intron here, I will not pick that up. However, if I do an RNA approach, I will pick it up because I'll pick up all these introns, I'll be able to sequence through that, and I'll be able to identify all the fusions that are present, not just the targetable ones. So again, it'll pick up intronic breakpoints and these novel ones, whereas the DNA won't. It'll also pick up low allele fractions. And also it supports the fact that if you've got it in the RNA, it's actually being transcribed, whereas the DNA, you're never quite sure. And the power of this can be seen in this particular data set here. So this is from Memorial Sloan Kettering. If you just look at the DNA test, you can see these particular cases in this oncoplot that have been picked up. If you add RNA in, you can see there's about another 15 to 20% of variants that have been picked up using the RNA approach as well as the DNA approach. So that's why in the recommendations and the guidelines, an RNA and DNA approach is recommended. So I think I'm out of time. I'm willing to take questions. I think they're going to go into the chat. Otherwise, I will hand over to Jen and Caroline, I believe. Thank you very much for your attention.